Let's continue our discussion of anti-neoplastics by focusing on drugs that affect microtubules. Let's refresh our memories on how microtubules are used in the cell cycle. Specifically, we're looking at mitosis. During mitosis, the cell develops and produces spindle fibers. And so here you see a visual representation of what may be prophase. The replicated chromosomes in metaphase are lined up down the middle. And then in anaphase, the microtubules that are making up the spindle fibers shorten and pull apart the replicated chromosomes. Once they're separated, the spindle fibers are no longer needed and the cell dissolves them or gets rid of them. So looking at this process, there's actually a couple of different places where our drugs can interfere. Let's start off with the Vinca alkaloids. And the Vinca alkaloids are actually working in the initial portion of this. They're preventing the assembly of the spindle fibers by preventing microtubules from coming together. The Vinca alkaloids originally come from the periwinkle plant. They're frequently referred to as spindle poisons. They bind to tubulin to prevent it from attaching to other tubulin subunits. And there are three drugs that are in this class: vincristine, vinblastine, and vinorelbine. Vincristine and vinblastine are very similar, used in fast-growing cancers such as acute leukemias, and in the case of vinblastine, testicular cancer. For vincristine, we also see it used in non-Hodgkin's lymphoma. In the acronym CHOP, it is actually the O, which stands for Oncovin, one of the potential brand names of vincristine. Mention that it's used in leukemias, specifically acute lymphocytic leukemia. And vincristine is also used in the treatment of multiple myeloma, which is a cancer that arises from plasma cells. Vincristine is primarily given as an intravenous bolus, but could also be administered as a continuous intravenous infusion, like when it's used in multiple myeloma. The dose-limiting toxicity of vincristine is neuropathies. That's something fairly unique to it. It doesn't cause myelosuppression to the same degree as other drugs do. In fact, when vincristine is used by itself, its effect on the marrow is minimal. You see other side effects that may be part of the neuropathy, including jaw pain. The pain will start usually at the insertion of the mandible in the temporal bone and then radiate down the jawline. Constipation is also a frequent side effect of vincristine as well. The constipation usually isn't something that's problematic because so many other chemotherapy agents that are used with vincristine can cause diarrhea, and so the effect is really canceled out. Mention that vinblastine is very similar to vincristine, and it has some overlap in therapeutic use, but it also has a couple of things that are unique to it, including Hodgkin's disease. It's part of the regimen ABVD, which is one of the standard regimens we use in the treatment of Hodgkin's lymphoma. It's also used in testicular cancer that has relapsed after initial therapy. That makes it a little bit unique over vincristine. It's given only as an intravenous bolus, and its dose-limiting toxicity is myelosuppression. Unlike vincristine, vinblastine is myelosuppressive on its own. You see other side effects. Once again, we're seeing neuropathies with vinblastine as well. Vinorelbine is different from the other two. You can tell based off its indication. It's used in the treatment of breast cancer and non-small cell lung cancer. Vinorelbine is also given as an IV infusion, and it also causes myelosuppression as its dose-limiting toxicity. It doesn't cause neuropathies to the same extent that vincristine or vinblastine do. You see some other side effects, and one that's unique is to it versus the other two drugs in the class is pancreatitis. Let's look at the taxanes now. The taxanes work by preventing the disassembly of the spindle fibers. So while Vincas are working early on in the process by preventing the microtubules from forming, your taxanes work late in the process and prevent them from being broken down. That means they remain stuck to the replicated chromosomes, and that keeps them from being able to function. Taxanes originally come from the yew tree, with paclitaxel, the first compound, coming from the Pacific yew tree, and docetaxel, the second one to be developed, coming from the European yew tree. The three primary drugs in this class are paclitaxel and docetaxel, and then the third drug that's fairly new, cabazitaxel.
Hello. Today we will be discussing the anatomy and physiology of the urinary system and the kidney. We will start broadly with the urinary system and then become more narrow and discuss the kidney, then the nephron, then the juxtaglomerular apparatus. Here is a view of the entire urinary system. We start with the kidneys, go down through the ureters, then into the bladder, and you can see the bladder is composed of bladder wall, the bladder lining, then the opening of the urethra, and the bladder outlet, and then finally to the urethra. So this is the flow of urine. So urine's created in the kidneys and then flows through the ureters into the bladder and then to the urethra and then is expelled. This is the broad picture of the urinary system. Now we'll focus more specifically on the kidney. This is a cross-section of the kidney. As you'll see, it is composed of an outer cortex and then the inner medulla, and then each of these triangular spots are renal pyramids, into the renal pelvis, and then in through to the ureter. So the renal pelvis leads into the ureter. And as you can see, we're going in through the renal artery. So the renal artery is leading into the kidney. So that is where the blood is brought into the kidney in order to be filtered. It goes in through the nephrons, which the nephrons are the individual functional unit of the kidney. As you can see over here, the nephrons go from the cortex through the medulla. And right here, you'll see up here, and we'll talk in a second a little bit more about the nephron specifically, but you'll see the majority of the nephron is actually up here in the cortex. So this is the glomerulus. So this is where the blood vessels are leading into, and where the glomerulus, where we filter the blood. And then it goes through the different parts of the nephron, again, primarily into the cortex. And what's primarily in the medulla is the loop of the nephron. But again, there are millions of nephrons within the kidney. So the nephron is the functional unit of the kidney. You have two kidneys. So you have several million nephrons within your body. They're not all functioning at once. So if there's death of one nephron, then there are others that can take over. So in a patient that has any type of renal injury or chronic kidney disease, the nephrons progressively lose function. However, there are other nephrons to take over that function. And once a patient develops some type of kidney disease, really you have a loss of already several nephrons before that's actually detected in any type of lab values, just because there are other nephrons that can take the place of the ones that die. So we really have several that die before you actually see a decrease in that creatinine clearance or the glomerular filtration rate. But back to our anatomy and physiology. Once we have the nephron filtering the fluid and waste from the body, that's filtered throughout the nephron and then into the collecting duct, which is the final part of the nephron, which we'll look at in just a second. Once that is done, it'll go into the renal pelvis and into the ureter. So to look more specifically at the nephron, so again, this is the functional unit of the kidney. We see we have a glomerulus right here. So we have an afferent arteriole that is bringing the blood to the nephron. That blood goes through the glomerulus, and that is where it's filtered, and then leaves through the efferent arteriole. So the blood that's filled with excess fluid and excess waste comes in through the afferent arteriole. It's filtered at the glomerulus, and then the blood that has been filtered leaves through the efferent arteriole and then back into systemic circulation. Where the glomerulus filters to is right here. That's the Bowman's capsule. From the Bowman's capsule, we get into the proximal convoluted tubule right here. And again, this is up here. All up here is in the renal cortex, so that kind of outer shell that we saw in the kidney. And then down here is the medulla, which is more of the inner part of the kidney. After the proximal convoluted tubule, we have the loop of Henle. We have first the descending loop of Henle, and then going down, and then coming back up is the ascending loop of Henle. And then after the loop of Henle, we get to the distal convoluted tubule, and the distal convoluted tubule leads into the collecting duct. So this is where the formation of urine occurs. So the waste and the extra fluid that is filtered from the glomerulus goes through the nephron. And in the nephron, we have a couple of different processes that occurring. So first we had filtration from the glomerulus, but in the nephron we'll have both reabsorption and secretion. So reabsorption is actually reabsorption back into the systemic circulation. So reabsorption of any electrolytes or fluid that's in the nephron back into the systemic circulation. And secretion is the opposite. Secretion would be secretion of electrolytes or other substances or fluid from the systemic circulation back into the nephron. So all of those processes together work to function to form urine. So the final product when it gets to the collecting duct is the formation of urine that again leads to those ureters, to the bladder and urethra, and is expelled.